So today I'm going to talk a little bit about reactive transport modeling in wetland systems. So we've been talking all week about how to use omics to really incorporate microbes into these models. And so I'm going to talk to you a little bit here about the specifics of and my thoughts about doing this within these wetland ecosystems. And so in some ways, my talk is going to be a little bit opposite of what you just saw, which I think presented a really great synthesis and synopsis of sort of a bigger picture way that these things matter. And I'm actually going to go into these systems, and I'm going to look at all the challenges that are present within these systems and some potential workarounds for those challenges as we move forward. But also because of the nature of the systems, the scale that I'm going to be talking about is much smaller as well. And I think that's sort of been a theme throughout the course is the importance of considering scale and what your question is in terms of how to apply and how to use these approaches. Okay. So wetlands is a broad category of ecosystems. You know, wetlands are really diverse systems that are found in freshwater inland wetlands that can be small and dry part of the time, or seasonally flooded, to sort of these permanently flooded coastal systems that have a lot of salt influence. And so even though, you know, we kind of all lump them together as wetlands, one sort of system, it's actually a bunch of different systems that each have different drivers and there are different considerations to be taken when we think about how we want to model and how we want to think about these systems. Hydrology is a really strong key in all of them, and it's what links it and makes it sort of a really great topic and system to consider in terms of integrating microbes into reactive transport models. But there are a lot of other key drivers as well. You know, whether or not the system is tidal, you know, is how constantly flooded is it? Is it seasonally flooded? Is it flooded year round? How much does the water depth vary? You know, what controls the water depth? Is it precipitation? Is it groundwater? Is it wind? You know, is there salt present or no salt present? What kind of vegetation is present, right? So when we talk about wetlands, we can talk about anything from sort of these herbaceous dominated systems to tree dominated systems, and those work and function in very different ways. But one thing that wetlands do tend to have in common is that they're where terrestrial areas meet water, right? And as such, they tend to be hot spots of biogeochemical activity because they have a mixture of sources of nutrients and other materials, and it's sort of the exchange between land and water. And so they tend to be hot spots, and so it's really important to capture what's happening within these systems. So like I said, I'm going to talk a little bit about the challenges, and I want to give you a little overview of what some of those challenges might be within these systems. And then I'm going to walk through a few case studies where I sort of show and demonstrate how these challenges are sort of affecting the system, and then also present some of the potential workarounds or adjustments to the approach that you've been shown that might help us incorporate this and really move forward in these important systems. So first off is, you know, community turnover along gradients, right? So we're talking about incorporating microbes into these models, but microbes aren't everywhere, right? And so a lot of the common gradients in sort of these wetland ecosystems, or these gradients and interfaces between terrestrial and aquatic, are also really strong drivers of microbial community composition and diversity, right? So I was talking about, like, in coastal systems, there's a really strong change in salinity. And in almost all of these systems, there's a change in flooding from the upland to the standing water, right? Where you get sort of this redox or oxic to anoxic gradient. And we know from lots of studies looking at sort of broad level biogeographical patterns in microbial community composition that salinity and redox are two of probably the top 3 factors that are really driving community composition. And so it's important to realize that as we move across these gradients, the microbes that are present in one location aren't necessarily going to be present in another. And so we need to think about how we want to incorporate that into the models. Another thing we need to think about is, and that we've talked about some, is the role of interspecific interactions, right? So this is one thing that's really unique to the biology aspect of biogeochemistry and these modeling approaches, is that in biology, life finds new and different ways to do everything, right? And so when we consider sort of these upland systems, or sort of mostly aerobic systems, you know, a single organism is capable of going from a polymer that's present in, for example, plant litter, using some extracellular enzymes, breaking it down to a monomer that can be taken up within the cell, and then doing all of the metabolic processing on that, and then releasing carbon dioxide into the atmosphere, right? So a single organism can actually go through all of these steps. Now, we know in natural systems that's not often always the case, and there are plenty of organisms that really specialize on degrading these polymers, and then other organisms that kind of cheat and come in and steal the monomers. So we know that it's not usually quite that simple, but we know that it also can be, that the metabolic potential to do that is present in individual organisms. But then when we start thinking about anaerobic degradation, there are far fewer organisms that can actually take a polymer, break it down into the monomer, and then bring that all the way down into carbon dioxide and methane, right? So here you tend to get fermentative organisms that can make a single change, and then they excrete a waste product that's then used by another organism for another small modification. And each of these reactions yields less thermodynamic energy, and there's tend to be slower growth for these organisms as well. And so that has potential impacts on how we want to model the microbes, because if you take here a single genome metabolic model, you're capturing one of multiple steps in this pathway. And so you have to consider whether, for your question, that's really if that's really useful, or if we need to come up with a way to make models, some community models that represent these groups of organisms. And then we need to consider how these groups of organisms are responding to those gradients. Another thing that is sort of a little bit swept under the rug, but I was very happy to see somebody ask directly a question about this in Discord earlier this week, is what about those quirky metabolisms, right? So metabolic modeling approaches, and it makes sense why, right? They've been developed for aerobic heterotrophic organisms. We started with E. coli, right? We started with these organisms that are easy to grow in the lab. We can study them and we can test things so we can understand what their metabolism is. And then we use those as the basis of making the annotations that you use that tell us which reactions are to put into the metabolic models. And most of these transformations are based off of the carbons and the atoms in the system. They're not based off of the protons and the electrons, which can sometimes go missing. You know, I'm sure you notice that you don't actually specify a pH in the conditions when you're looking at these metabolic models. And so for that reason, some protons and electrons can kind of get mixed up a little bit. And that poses a problem for metabolisms that really rely on the extracellular electron transfer. Now, Chris Henry and his group are doing a lot right now to be able to improve the capture of these things. But there's still it's still sort of a gray area, and something we need to consider. In addition to that, just by the very nature of being less well studied, these metabolisms tend to have more misannotations when you're trying to figure out what's in the genome, and fewer reactions that then get included into the draft models, et cetera. So the draft models themselves simply require more curation, right? So now I'm going to sort of walk you through some of these example systems and studies that show that these things are relatively common in these wetland ecosystems, but also how we can work around it, how we can keep moving forward and keep advancing this really exciting idea and approach that we've developed and we've been talking to you guys about today, through this past week in the course. So my collaborators on this project, just want to give some quick announcements on that. There's Loretta Battaglia, who's now the president of the Society of Wetland Scientists, as well as Julia Terry. And so this is a study that we conducted down at Grand Bay National Estuary and Research Reserve, which is on the Gulf Coast in the state of Mississippi. So here's a picture of the site, and you can see this transition from the Gulf on one side to pine trees, which are not particularly fond of flooding or salt, within just the scope of this picture, right? And so the gradient that we're talking about here is 100 centimeters in elevation and less than a kilometer in distance. So if you'll think to the talk we just heard about the scale of grids and how we do these models in the system, this is really relatively small. And along these gradients, we see that there are 4 different plant communities. You can't capture all of them in the picture here, but there's the pines here, there's salt-tolerant grasses here, there's a mixture here, which you can kind of see the taller grass, of brackish marsh species, so a little bit more diversity. And then in between, which you can't really see in the picture, is a fresh marsh, which has a lot more species, a lot more diversity in this system. Right? And so for this, we collected samples from 4 transects on 4 different what we call pine islands, because the islands the pines themselves are sort of surrounded in a sea of these marshes, not because they're surrounded by water. And looking at the microbial community composition, we see that there's a very strong gradient between these different sort of plant communities here in terms of the microbial community composition. And this has been restructured so that the maximum variation explained in the variation in the community is along this x-axis, right? And so you see that this is pretty much a straight line along that axis as well. So the strongest driver here, sort of these overlapping flooding and salinity gradients that very small change in elevation represents. And then I want to show you here, this Venn diagram shows the number of unique taxa that are present in each sort of combination or, in an individual site along that transect. And what we see is that the majority of the species are found at only one of these locations, and a very small number of species, less than 1.5% of the total number of species that we found at this site, are present across the entire site, right? And so we're talking about microbes. They can move, they can be dispersed very easily. There's a lot of water transfer. There are a lot of animal movements through this really short gradient. But what we see is that the community almost completely turns over between the salt marsh and the pine woods in these islands. Right? And so when we're talking about community turnover, it's a really strong, sharp turnover. And this isn't just, present there. This is actually a really common pattern. And so I found that same pattern in studies that I've done in Weeks Bay, Alabama, which is a very similar system, but also at Elgin Air Force Base out in Florida. And in this, it's along a river that has a salinity gradient in it as well. And so samples were collected from wetlands along the length of this river. And it's not just limited to the Gulf Coast either. From some of the work that some of my collaborators have done, I see this out in the Pacific Northwest as well. With the overlay of salinity and anoxia, you really get these strong gradients, right? So what does that mean for reactive transport models? Who knows? We haven't started incorporating microbes in, and so this is still really unknown territory. But if the functional redundancy is high, and Christoph alluded to this in his talk earlier, the fact that there's community turnover may not actually pose too much of a problem for our ability to model what's happening here, but it's still something we need to consider as we're starting to incorporate genomes into these models. But also, if the community turnover is closely linked to the chemical turnover, so for example, in a coastal system, sulfate's going to be present in seawater, but not in those freshwater sites. So if you just lose sulfate reducers where there's no sulfate, that's not really going to affect the performance of the model either. But if those aren't the case, it may also be possible to deal with this using different machine learning approaches, right? So if I look at the actual distribution of individual OTUs across this, and these are sort of the average point for each of those different zones, you'll see that, they clearly sort of show a preference in general, and they don't all have the same preferences. But when I looked at from the subset of less than 1.5% of the taxa, 90% of those that were present across the whole gradient showed this sort of a zone preference, right? And so it'd be possible to use machine learning approaches and not black box approaches like the neural networks that Hyun was talking to us about earlier, but white or gray box machine learning approaches. And so what that means is that they can actually give you the underlying equations for the relationships that explain the distribution of these taxa. Then we can incorporate those relationships into the reactive transport models in a similar way to how we'd consider other external factors, like Roloff was explaining yesterday in terms of how pH might control the accumulation of biomass in the system. Right, so the community turnover is a challenge. It might be something that really affects us, but we have some alternative approaches, and there are other ways and other things to consider as we move forward. It's just something to keep in mind, right? So the next system I'm going to talk about is one that's maybe not quite as strong in terms of the gradients, but where we still see some real differences in terms of what's happening here. And here in particular, I want to focus in on the interactions. So here are my collaborators for this project. It was a slightly larger project with more people. The study site is one of the wetlands here at Argonne. There are more than 30 wetlands on site, and I study most of them for various reasons at different times. But this is a permanently flooded inland wetland. It has a water level control structure, so the water level tends to be pretty constant. It does drop a little bit more in the summer when there's just not enough water because of evapotranspiration in the area. It receives some treated lab wastewater, so it's a little bit nutrient heavy, but it's been treated, so it's not too bad. We collected water samples in the top 5 centimeters of sediment roughly once a week for 50 weeks. And so in terms of the community composition, looking at this, we see that it's relatively constant through time, right? So we're going to the same spot, we have relatively constant water depth. The other conditions are relatively similar as well. We still have changing seasonality and things. And so what we see is, especially for the sediment, but somewhat also for the water, we have relatively consistent community composition. Great, that helps us model things. We don't have the same problem that I was just discussing. But there's a difference between the composition and the interactions of community members, right? And so I went ahead and I looked at the interactions that were present in these soil microbial communities. So I focused in on the sediments here. And I generated 2 types of networks. So I generated a single network that explained all of the data. And then I also looked at sort of 43 networks that were based on a sliding scale. So like week 1 through week 8, week 2 through week 9, etc. And then sort of compared what happens as we look through it. So if we look at the interactions through time, what we see is that they change quite a bit. Right? So although the community composition itself is relatively stable, potential interactions between community members, the activity, what they're doing, changes through time. And I'm not actually going to show the whole video here because even though it's only 2 seconds, it does seem to take forever. But this is right here where I wanted to show you what happens, right? So initially you had sort of this large ball of interactions where most of the species were interacting with each other. And then that kind of fell apart for a while and you had sort of these smaller groups of species interacting, right? And then as we go through, it kind of returns to where it was before. So that's sort of the highlight of that. But if we look at sort of these interactions, across all of the different networks, there were more than 60,000 unique interactions. But the vast majority of those, even though I'm using a sliding scale, so most of the data from one of those plots to the next is exactly the same, right? So 1 to 9, 1 to 8, 2 to 9. So most of those overlap. 46,000, 72% of those were only found in a single network. Right? And so we know that these species are sort of, can be interacting with each other in a really flexible sort of way. And then, but there were some that were really common and that were present in a good majority of the networks. And some of those were really interesting, right? So most of them were phylogenetically similar taxa. So maybe it was just the environmental conditions, the microbes were changing in the same direction with the environmental conditions. But one that was particularly interesting was that there was an Ignavibacteriaceae, which is a primary fermenting organism, and then a geobacter, right? Which uses primary fermentation products. And so there's a reason to believe that that's sort of a metabolic interaction, and it's one that could really influence how iron is being transformed in the system and really link iron to the carbon that's present, and not just to the carbon that's present, but also to which species is using that carbon that's present. And so that's something to keep in mind as we are sort of moving forward. And another thing that was particularly interesting in this, so as we look at this sort of big picture of these interactions, right, across all of the different the sampling points, we make a single network. You know, we have just under 4,000 taxa. So under a quarter of the taxa that are present in the entire system were interacting. We only have 12,000 interactions here. So most of those that didn't show up in those sliding scales also don't show up here. But we have 113 modules, which if you've looked at interaction networks from other microbial ecosystems, specifically sort of the uplands ecosystems, you tend to see one giant ball. All of the organisms are kind of interacting with each other to differing extents, but more or less loosely interacting with each other. Whereas here you see a lot of clusters that sort of break off and are interacting with each other. And so these consortia then are really only interacting with each other. And there seem to be a lot of them that are similar size and similar shape. And so, you know, if you think back to what I was saying earlier about the interactions in the system and how degradation of carbon happens, that it's a multi-step process involving many organisms, you can imagine. And it hasn't been shown, right? Because this is only 16S data, and I haven't had the chance to go in and try to find all of the genomes of all of these species to see if this theory makes sense. But it's easy to imagine that these taxa represent groups of interacting organisms based on that exchange of metabolites. And so if that's the case, the question really here is, with these small clusters, with these consortia of organisms, you know, how do we model the change from one consortia to another? What sort of an effect does that have on the overall transformation of elements in the system, or how that transformation at a location changes with environmental conditions? And one of the great things about these reactive transport models is it really allows you to look at sort of how physical and chemical and biological changes are all interacting with each other. And so the question here really becomes like, how do these, this biology, these different consortia, do they respond in the same way or different ways to changes in the environmental condition? Because that affects how we want to model it and what we need to be thinking about when we try to incorporate it into models. Right? And so there are plenty of examples in the environment of how biology can outwit thermodynamics, right? And so, you know, we used to think, and Christoph showed this sort of pretty well earlier, how, you know, sulfate reduction happens and then sulfate reduction stops and then methanogenesis starts because methanogenesis doesn't yield as much energy as sulfate reduction. It's not as thermodynamically favorable. And yet we've found that actually those things can and do co-occur. And interestingly, you know, recent work has shown that methanogenesis in oxygenated soils is a substantial fraction of the methane emissions from wetlands, right? And so methanogenesis wasn't even thought until relatively recently to be able to occur in oxygenated systems, in part because a lot of the methanogens we knew about were very sensitive to oxygen, right? They were not tolerant of oxygen at all. And so it's still possible for that to happen. And you know, there's been some studies, and this is only bioarchive, right? So maybe hold your breath a little bit, but cyanobacteria in both aquatic and terrestrial environments can also contribute to the formation of methane. And there's many, many examples of sort of where this paired metabolism allows processes to occur under otherwise unfavorable conditions. And so while using thermodynamics to model and to pick ideal pathways for what sort of processes are happening in a reactive transport model really makes a lot of sense. And the majority of the time, the vast majority of the time, is probably perfectly accurate. Once we move into these systems where there's sort of more variability and redox, and where the thermodynamics situation is a little bit different, it's something that we need to consider and examine a little bit more closely what those assumptions mean, and more importantly, how those assumptions affect our models, right? So it's not something that should stop us from using the thermodynamic approach, but it's something that we should pay attention to when we're evaluating that model and how we discuss that model. Right. So overall, what are this sort of abundance of interactions mean, and how does this change, right? So for community turnover, you know, when we're in sort of this more stable system, you know, it's less likely to be important, you know, as we're sort of changing through time, but it's not really very much. And even when we do see the changes, there's a greater probability of functional redundancy because the, you know, environmental, the physical and chemical conditions are more similar, you know, and should it pose a problem here, we could sort of employ some of the same adaptive approaches I described before. But for the interactions, there's a lot more questions. There's a lot less known about what's actually happening in the environment in terms of interactions. You know, this is one of the big unknowns right now in microbial ecology, is how common are interactions in the environment, and how important are they for, you know, understanding how microbes are impacting ecosystem processes, right? And so one thing that we can sort of approach we could take here is doing some of the pore scale modeling that Christoph was talking about. Okay, so if we do this really detailed, you know, this more detailed modeling where we can kind of get at these interactions, and we can put different species, you know, spatially located differently, and we can see how metabolic exchanges are happening between them, and the sort of the time scales that's happening on, or more reasonable time scales to how quickly that's happening. How does that impact what happens at the bigger scale, right? And so how does that spatial distribution at the micro scale matter for the macro scale? And how redundant are these consortia? You know, is it having a big impact on what happens? And can we substitute in one consortia for another in these models? And then what's sort of the sensitivity of that? How much does it change our model output, right? And depending on the results from that, we may need to develop approaches to generalize and to combine these things, to bend them, if you will, into different sort of groups, so that we can still use these modeling approaches and really understand how microbes are impacting the system. But this is sort of one of those great places where the MODEX approach really comes in handy, right? So let's, you know, go ahead and make some microbial transport models. Let's see what happens and see how it compares to reality, and see where we need to make some of these adjustments. And so, you know, those were a couple of examples, and I'm going to go through here maybe just one more. These will be sort of a couple of little brief vignettes, really, from the work that I am doing with the Argonne Wetland Hydrobiogeochemistry SFA. This project is sort of very recently started, so we just got a new field site. We're adjusting everything to that, and then you know, with COVID and everything, we haven't been able to get out there very much, and so there's a little bit less data here, but I still want to talk a little about what we're doing and sort of how that ties in with the ability to sort of use this sort of approach to incorporate microbes into reactive transport models. So here's my collaborators for that project. Okay, so the study site for this is the Thames Branch, which is a creek at the Savannah River site. Right? I don't know how familiar you guys are with DOE sites, but the Savannah River site is heavily impacted with legacy waste. There's, our field site includes both a control and a contaminated area, where the contaminated area is very strongly impacted by some depleted uranium releases, as well as heavy metals, right? And so these have had a really big impact on the environment, but it also makes it very important that we understand with really good detail what's going on in this system, right? So the Thames Branch empties into 3 Mile Run, which empties into the Savannah River, which the good people of Georgia use as drinking water. And so it's important for us to understand what's happening, what transformations are happening with the uranium at various levels, in order to keep water quality high, and for us to understand what's happening with that. And so as part of the SFA, we have established multiple sites along the creek, and these are in sort of ecologically distinct areas, and we've, as much as possible, tried to control those in the contaminated and uncontaminated zones, and they encapsulate both sort of the stream and stream bed as well as the wetlands on the sides of the stream bed. Yeah. And so a brief study that I did, just sort of trying to characterize the microbial community at the site. I sampled, took soil samples, again, sort of at the very shallow depth, 0 to 5 centimeters, throughout the watershed. And I focused on differences in the ecological parameters in order to sort of focus on this. And so some of those differences included, it being the contaminated versus uncontaminated sites, disturbances. There are a lot of disturbances in this forest. It's a relatively large and natural forest. There's a lot of wild boar, though, that like to dig and make wallows. So we have some wallows included in that, for example. Different plant communities. So some areas are herbaceous dominated and some are tree dominated. A sloping aspect, so right near Thames Branch, it tends to be relatively flat, and we get sort of some of these formations of wetlands, but it does rise. This is sort of the foothills of the Appalachians, so it's nothing that somebody from the Rocky Mountains would even call a hill, probably, but to people from the Delta in Louisiana and Georgia and those systems that I was just telling you about, that it seems like there's quite a bit of topography there, as well as sort of the position along the creek. Okay, and so one of the things I wanted to pull out here in particular is sort of an even smaller scale version of what I was talking about with Grand Bay, which is looking at hummocks and hollows. So sort of just these raised areas at the base of trees, and sort of lowered areas in between tree trunks that occur typically in sort of these southeastern wetlands. And so just these differences in topography, like we're talking less than 10 centimeters, probably most of the time less than 5 centimeters of difference in elevation between these locations. And these were all paired samples. Can result in this difference, the separation of communities. And it's a separation of communities that you can see here in spite of all of the other variation, right? So these things weren't pushed together because they're underneath the same plant species, or because they have very similar hydrologic regime, or they're in a similar part of, the similar location along the creek, right? There's this particular set of hummocks and hollows is from the control site. So that had control versus heavy metal contamination between the disturbance of a pig wallow. This actually still comes out as one of the stronger drivers. And again, this ordination is spun so that the maximum variation is along this XY axis, right? And so we're talking about these really small changes in microtopography, something that you're unlikely to capture when making sort of these larger scale reactive transport models, are really having a difference in what community members are there. Now, the other thing that we're really focused on here at the site, we're looking at the formation of iron oxide flocks, right? And so one of the goals for this is to sort of model the effects that these flocks are having on uranium transport. And because we went out to the site, sometimes these flocks are present, sometimes they're not. They're really discrete in terms of their temporal occurrence, as well as their spatial occurrence. But the samples that we took from them contained particularly high concentrations of uranium relative to sort of the background sediments in the area and the water itself. And so we think that these are probably pretty important for understanding what's going on with uranium and water quality in general in the system. But the challenge to try to create a model where we're looking at, both the microbial aspect of the formation of the flock and what's the transformations happening in the flock, obviously, as well as sort of the flow, water flow is going to impact when the flocks are forming and when they're not, in the chemistry of the system, is that they're really actually taxonomically and metabolically diverse mats. So when we looked at the community composition here, we found iron oxidizers, of course, but we also found some methanogens and some methanotrophs and some sulfate reducers, and a whole slew of different types of organisms that are doing different things within what appears to be a very iron-rich, a uniformly iron-rich microbial mat. Right. Another challenge with looking and modeling iron oxidation is that it can occur both biotically and abiotically, and sort of parsing that out is really important in order to capture it in the model. And even to add to that challenge even more, iron oxidizing metabolism and physiology is pretty poorly studied. It's not super well known. If you take a genome and you try to annotate it for iron oxidizing genes, you're going to run into a lot of trouble. And so if we were to take a genome from this and to try to plug it into K-base and make that draft metabolic model, we'd get a draft metabolic model out, but the chances that it's going to actually oxidize iron are very small, right? And that's one of the challenges of working with this metabolism. It's not sort of a downside to K-base or something that K-base can't do that other things can do. It's that actually making metabolic models of some of these metabolisms is a really challenging task. And so for that, the best potential solution that I could come up with is collaborate with somebody who knows what they're doing here, right? And so, I'm working on a project in collaboration with someone who's really deeply studied iron oxidation. Right, so she knows how to try to get at the kinetics of the iron oxidation and distinguishing it between biotic and abiotic, as well as having a good understanding of the metabolism, right? And so by doing this, by taking this sort of focused approach, I think that can really improve our ability to make models of this, but it's not something that's sort of a 1 week we can sit down and go from having some omics data to having sort of a rough way to integrate it with the reactive transport model. It's something that was going to require a lot more time, a lot more knowledge, a lot more curation to develop. That's certainly not a problem. It doesn't stop anything from happening, but it is the challenge in terms of moving this forward and getting things done. So with that, I just want to kind of do a brief recap here. So at the summer school, you've been exposed to this workflow. You've gotten some hands-on experience, at least some of you, with the approaches that you can use. And I think that this is an exciting way to sort of link all of these different omics data and to improve our ability to make good predictions with reactive transport models. I really want to, I'm sure you guys picked up on it, but in case it hasn't clear at this point, I really want to acknowledge Hyun's role in developing a lot of this workflow conceptually, as well as doing a lot of the work. You know, it's why he gave so many talks and did such a great job with that. You know, I'm really excited about this, and I think it's gonna, you know, really lead to some advances, and that more often than not, in most systems, for most questions, using this approach is going to be a really great way to move forward. But in some situations and some conditions, and depending on your question, some adjustments may be necessary. And so I wanted to talk a little bit about you know, some of the things that I could foresee. And, you know, a lot of you had asked questions sort of around these lines about, well, but what if? And those what ifs, I think, are really where the interesting changes and adjustments are needed, you know. And so just sort of to recap that, so with the turnover along the gradients, you know, some of the things that we can do here are to incorporate maybe some different machine learning approaches to try to capture how the microbial distribution is changing along the gradient. For the interspecific interactions, you know, some of the things we can do are sort of look at these pore scale models and see if we can use those to sort of incorporate the differences and how that's happening in different locations, and to do some of that scaling, as well as to sort of understand the impacts of and functional redundancy in different consortia. And then for the sort of the quirky metabolisms, the things that we don't know very well yet, or the things that, you know, have some of these weird electron issues, the best thing we can do is just sort of iterate with the curations, find the people, the physiologists. Physiologists always get overlooked in things like this, who really know the systems really well, and tap into the existing knowledge, as well as try to build knowledge in order to improve our ability and other people's abilities to automate this and to advance it moving forward. So as you embark on the journey of trying to use some of the skills that you've learned this week in this course, you know, you remember that, you know, as always, you need to match the question and the data and the model. And that's going to be sort of very system and very question dependent how you're able to do that. But it's going to be possible. Even if you have challenges, there's always going to be workarounds. And so don't let hitting the wall once to stop you from moving forward. You know, this is a very cutting edge area. There's going to be a lot of bumps along the way. Some of them we might be able to foresee and kind of plan out, and some of them we won't be able to. But, you know, I think that this is really going to, these approaches are going to really be able to improve the predictive ability of these reactive transport models. But also, in addition to just improving that, it's going to help us both the adjustments that we need to make, considering what those are, as well as just iterating on the model and experiment design, can tell us a lot about the components of the models themselves. And so here I've been talking a lot about sort of the microbial ecology of that. And so this can tell us a lot about sort of what things we don't know. For example, you know, right now we don't really know how common interactions are in the environment or how much they matter for ecosystem processes. One really great way of trying to figure that out is to take what we know now and try to put those microbes into these reactor transport model frameworks and see how big of an impact it makes. See what the sensitivity of those models is to changing those microbial parameters. And we can really sort of start to explore in a more controlled and guided way, using MODEX, sort of how that happens, how important are interactions, what are the effects that they're having, and where are they having those effects. And so lastly, I just want to thank all of you. It's been really exciting to me to see and hear your enthusiasm and your excitement about doing this. And I think that we're really developing a strong community here that can work to develop these approaches. And yeah, I guess I have maybe a couple minutes for questions or whatever you want to do, Tim.
Yeah, we've got some time for some discussion. Thank you, Pamela. That was a great way to wrap up the workshop. And there's a lot of interest. Well, so one thing is, like Xingyan, people were asking if you have some papers that you can share. How to add papers to the Discord messages is apparently a little tricky. I tried to add one, and it said it was too big. So if anybody has some help over there in Discord can suggest how to add paper links, that would be helpful. But actually, there was a whole bunch of discussion around and interest in your interaction diagrams. And so I'm gonna, there was kind of two threads of questions around that. One has to do with sort of what underlies them. So, really what does interaction actually mean? What are the data that you're using to provide interaction information? How do you actually determine interactions? And then the second thread is kind of what are the software that you use and how do you actually implement that?
Okay, so I'll answer them in the thread order that you presented there. So in this case, and in most cases, I think the interaction networks are derived from sort of 16S data, which it really captures sort of the taxonomic abundance of organisms. It does that in a perhaps more straightforward and in-depth way than a lot of metagenomics approaches can. So shotgun metagenomics is great in that it tells you all of these genes and all of these functions. But when you're interested in whose they are focusing on the 16S rDNA gene, the ribosomal gene, which is what we as microbial ecologists use to classify organisms, focusing in on that target gene really gives you much better resolution, and you can get much better depth, and it also happens to be a lot cheaper, so you can take a lot more samples. So it kind of works well for all of those reasons. And so in considering what makes something an interaction, what you do is you take samples, and in this case, I try to limit it to no fewer than 8. That's just sort of my personal cutoff on what I think is reasonable. But you look at whether or not the species are changing in ways that would indicate that they might be interacting, right? So do the species abundance go up together? Do they go in opposite directions? Do they go down together? And for doing this, a lot of people, and there are a number of approaches, and I'll post a paper that kind of gives an overview of many of them, that just use sort of correlation-based approaches. But in this instance, what I've done to try to capture some non-linearity is use mutual information, which allows you to say, knowing the relative abundance of A, this is the relative abundance of B, how much do I know about that? And then, B to A as well. But then even just doing that, you get a lot of false positives. Like this method is very much known to have a lot of false positives. Things aren't actually interacting, they just are apparently interacting. And that explains probably a lot of that, oh, well, it appeared once in that series of sliding windows, right? Is that correlations happen by chance. So what I've done is I've developed a random matrix theory-based approach, which looks at sort of the distribution of the significance of those interactions to try to tease out signal from noise based on those distributions. And that's just sort of at least mostly objective approach as to where to draw the cutoffs. A lot of researchers will just set a cutoff of a super low p-value in order to throw out a lot of the false positives, or only take the top 100 strongest interactions and look at those. But my approach has been to try to use sort of this objective cutoff, and then, as you saw, to compare multiple different interaction networks to each other and see which interactions are consistent. Because if there's an interaction that's consistently happening, it may not always be the strongest, but then it'll still appear there. So that's sort of what I've used there. It doesn't necessarily mean that the species are in fact interacting. And so what you need to do that is to pull out other information on the species, like I did with the Ignavobacteriaceae and the Geobacter. We know enough about those that we can hypothesize what that interaction would look like, and so you could feel good that those are interacting. Whereas the ones that I didn't label, and I mentioned this earlier, were phylogenetically similar. So they might just be not very far evolutionarily diverged and responding to the environment in the same way that gives that sort of apparent pattern. And there isn't right now a clear way to distinguish between those things. What's happening in terms of environmental filtering versus when the organisms are interacting, besides diving a little bit deeper. As far as software, just sort of keeping an eye on time here too. You know, I'll post in there a review, sort of an overview paper that I've written that describes a few of the different ways to deal with that. But I'll also sort of put in a plug here for KBase. I'm actually currently implementing my approach in KBase, as well as a lot of other tools and ways to look at this 16S data in KBase and do some of these analyses. And so that will also be available in KBase hopefully soon. I have a student who's trying to get it done for a summer project in the next couple of weeks. So we'll see if that actually happens. It'll probably take a little tweaking after that, but it's coming together pretty nicely in terms of getting the app designed and made. So